we met already quite a long time ago. I think it's 20 years or 21 years ago, or so, maybe more. So we were younger than that at the time as well.
Oh, yeah. Thanks, Bruno.
And and as well at the time, the kind of cool thing was quantum physics and quantum computers, and so that's what we were working with. And whereas like at the conference the other day, I realized that there were like there were 3 or 4 of us actually who come from the quantum physics background, who are now more in the imaging and biology side of things. And so the first question is for you, was it like an intentional shift, or did it kind of happen?
It's a bit of both, I suppose. So it's, the unfortunate, or one of the unfortunate things about academia is, when you're a postdoc, they're short-term contracts, and you're always kind of, looking for the next job, and you have some element of luck in what's available at different times. And so I was I was coming to the end of a contract up in Leeds, and I was looking around for other physics jobs, similar physics jobs as that, but I was also keeping an eye on other options. I've always been sort of interested in a range of things, and I was kind of emailing friends that I knew from various times during studies, and that I worked with in the past. And I emailed somebody called Katie Bentley, who I did my MSc with. So we did the MSc in Evolutionary and Adaptive Systems at the University of Sussex in about 2001-ish, something like that. So she did maths undergrad, computer science masters, and PhD, and then she'd ended up working at Cancer Research UK, the London Research Institute, and we were sort of in touch on Facebook or something, and I kind of just sent a speculative message, any interesting jobs for physicists around there. And it just happened that her and her boss had been to Germany, I don't know, a few weeks before, to look at one of these amazing new microscopes. Actually, it was a light sheet microscope. So I know one of your previous interviews was sort of around this sort of light sheet microscopy area. So and this was, I guess, around 2010, something like that. They were sort of really enthused about having a physicist who could maybe build stuff, and also image analysis was, has always been a major problem, and having people who can do sort of custom coding in biology outside of the normal sort of bioinformatics is a bit rare. So it was quite hard to build the sort of analysis pipelines that they really wanted to do. So yeah, I sort of got in through the side door at CRUK, and I sort of had a very nice introduction to the team, and we chatted about what might be possible. And yeah, it was a real lucky move. I happened to message somebody at exactly the right time, but it sort of really defined a whole kind of career direction for me.
And so, and has your physics background, because of course there's a lot of physics in optics and also in electron microscopy. So has your physics background actually helped you for the imaging part of things?
Yeah, definitely. So I don't do so much of the actual imaging myself, but actually my main contributions at the moment. So I joined Cancer Research UK in one of the research groups, but then the group leader, Holger Gerhardt, he moved to Germany, and then I moved over to the Electron Microscopy core facility at Cancer Research UK, which is now migrated into the Francis Crick Institute, which is where I am now. And the sorts of things I've been doing in the electron microscopy team have been around designing hardware to do imaging, a particular type of imaging called correlative light and electron microscopy. So we kind of combine fluorescence and electron microscopes together. And again, it was I suppose, a lucky piece of timing that, you know, my boss was about to leave to Germany and I wasn't really in a position to be able to go. And then I went to a talk which my current boss had organised. So Lucy Collinson is my current boss then at CRUK, but now at the Crick. And she'd organised a talk for an external speaker to come in to talk about this thing that I'd never heard of, correlative light electron microscopy. And they were talking about, you know, building hardware to do all these clever things. And I kind of looked at it and thought, well, that looks quite similar to what I did in my PhD. So it was big vacuum chamber. So electron microscopes all in a vacuum chamber. There was getting light down to a sample somewhere in the depths of this vacuum chamber. So, you know, we had tiny bits of space, so, you know, a few millimeters gaps here and there, and we had to pipe things in. And that's actually pretty similar to one of the main elements of the hardware development I was involved in my PhD. And I just kind of emailed Lucy and said, I have an idea. I reckon it would be possible to do this, this, and this. And then she replied and said, yeah, come downstairs and let's talk. And then, you know, I ended up joining the team that way. So yeah, in that sense, the sort of knowing the physics experimental methods you would use to perform a certain task, I think that was a real benefit. And then, yeah, like you say, all of these imaging things are subject to the laws of physics. So you have diffraction limits, and you have, you know, a particular size lens has a particular ability to capture light and so on. So all of these things are quite, you deal with a lot in experimental physics, but maybe not so much in experimental biology. So I think that was really a useful thing. And the kind of engineering side, so knowing the difference between the different cameras, you know, the quantum efficiency of the cameras and all of these kind of technical details which might not be very obvious what the benefits of different things might be to somebody who hasn't worked with these sorts of devices before. I think that was a real a useful thing to be able to contribute things I knew about that side of things. And then, yeah, just generally, I've always, in physics, I always kind of tried to straddle theory and experiment. So I didn't like this idea of kind of being pigeonholed into one or the other, which, you know, you sometimes get these two camps. And in my previously my master's and my PhD, I tried to use some of these adaptive computation methods to solve certain problems. And yeah, and as AI has come into image analysis in particular a lot recently, then, you know, I've been dealing with systems like that for quite a few years. And so knowing the backgrounds to that and the sort of pros and cons of those things, I think has really helped in a lot of the work I'm doing at the moment.
Yes, I do remember us talking like a long time ago, yeah, more than 20 years ago, about these adaptive systems, which were an AI, you know, basic AI systems for the time. And at the time, I also remember that we, and a lot of people chose to go, instead of like furthering research in that field, to further research in quantum computing and quantum physics, because indeed it was evolving at quite a slow pace at the time. But then, like, I have the impression that a few years ago, maybe 10 years ago or so, the speed at which AI has started evolving, like, overtook the quantum physics kind of evolution speeds. And so on the other hand, it could be that just, you know, probably we are quite excited about everything. So I was wondering if you're just like really excited about AI right now, and what's actually just to have your view on that, if, you know, if AI is really the real thing and if it really will help to solve some real problems. That'd be really, really interesting to know.
Yeah, so I think an important distinction to make, so AI is used, the phrase AI is used a lot and it means different things to different people. So broadly speaking,
Famous quantum.
Well, yeah, exactly. Yeah, add AI to anything, and you get extra money, right? That's how the funding works. Um, Not really. Uh, it's yeah, you can kind of look at it in two ways. So one, maybe the original idea about AI, back in the days of cybernetics and things like that, it was to sort of try and understand how brains work, right? So really, what is intelligence? How does it work? And that's clearly a very complex thing, and we're very far from really understanding that. I would say. I mean, I'm not a specialist in that, but it feels like these kind of what might some people call artificial general intelligence and so on. So these kind of systems that somehow are able to inform us in how brains work. It's an open question how close we are to that. But the other aspect, which is, I guess, more what people are familiar with, is a sort of more engineering side. So, I have a problem, and I have some data, and I have this. I want to get to this solution. I want to optimize this thing, and then it's, you can kind of do that via this kind of iterative optimization process. Just keep making it better and better and better until it gets hopefully good enough for what you need. But in that process, maybe you leave to one side the sort of understanding of what that thing's doing. So interpretability of AI is a very hot topic at the moment, and often it can be quite hard to understand what the systems are doing. But in the engineering sense of solving this problem in a way that I hopefully convince myself is robust and not open to various errors or artifacts, then that's a perfectly valid use, as any research tool to get to an answer in any question you're asking. And in the last 5 years in particular, but more than that, generally, the kind of particularly convolutional neural networks, deep learning methods coming into bioimage analysis has really revolutionized the field. I think where things had been moving quite slowly in terms of how quickly methods were improving, I think that's really made a bit of a step change in the last few years, and so things that were effectively impossible before are now starting to be possible, and in some cases even routine. And yeah, I think that's very exciting that it's some of the, so this background behind me is we applied a deep learning algorithm. So we have an amazing scientific computing team at the Crick. So Luke and Joost, who sort of wrote the system that did this. So this is a huge chunk of electron microscopy data that's been imaged over, I don't know, probably days or weeks. Hundreds of gigabytes raw data, I expect, and these objects, these colourful objects behind me, each different colour is a different mitochondrion from within a piece of tissue. So I think it's a piece of brain tissue, and so the mitochondria in lots of different cells, and there are thousands of them in this image, and you see that they're kind of all really nice and clean, and it's kind of astounding, having tried to do this with non-deep learning methods in the past, and you can't do it really. It doesn't really work, and it's I think one of the most exciting things about AI is that it's sort of cumulative as well. So I build a system that does this, and somebody has a slightly different data set. They don't need to start from scratch. They can take that system, do a little bit of work, and now it will work on their data. Whereas the sort of more traditional methods, you have to dig into the code, and you have to change all sorts of parameters yourself. And this kind of hand tuning is a bit of a dark art, and it's quite difficult to maybe justify what you're doing. Whereas if you let the machine, it's just an optimization problem, then I think it's a very exciting area for AI.
But it looks like if I were to have to label that data by hand myself, it'd be really difficult. So has AI actually overtaken what humans can do, like not just in the volume of how much data you can do it on, but on actually like having a really complex image analysis.
So yeah, I would say so. There is a sleight of hand which I've glossed over here, but at some point for this data set, so this was actually a competition. It's one of these conference competition data sets that people put out, and somebody in the team who made the competition has sat down and manually annotated this, or probably several people manually gone through this to provide the training data that allows us to build the network that performs the analysis, and then to compare against. Because of course, I could show you this picture and say, yeah, it's worked really well, but without checking, without validating that this is good, then you just have to take my word for that, and that's not how we do science, right? So this these supervised methods, so I guess I've been implicitly talking about supervised methods. At the moment, you still need fairly significant quantities of manually annotated sort of ground truth data to train the systems, but the hope is that as you bootstrap different methods, and you can use things like transfer learning, where you can use a smaller amount of data to adjust an already existing neural network model. I think that's really the sort of the next steps in the field, is trying to move away from this having to have huge amounts of manual annotations. But actually, before we move on, I'll just one thing that we've tried for trying to alleviate this bottleneck of somebody having to sit down and manually annotate all of these objects. Another thing that, as a physicist, I was sort of aware of many years ago, I think 2007 something that started, there was a really fascinating project called Galaxy Zoo, which is a citizen science project, and astronomers have a similar problem to microscopists, so they can generate millions of images really easily now, and just tons and tons of data, and it's the throughput of the analysis is too low to deal with it. So these the Galaxy Zoo team, led by Chris Lintop, they came up with this this method for kind of cropping images and sharing them out on the internet, and then you explain to people, this is a scientific project. If you want to help us, come along and help us annotate these images. And it worked really amazingly well in Galaxy Zoo. And then, so a few years ago, maybe 5 years ago, maybe a bit less, we set up our own citizen science project where we use this sort of power of the crowd to help us generate the ground truth. So not for this data in particular, but for
this HSL, is it?
This is HSL, yeah. So we have the HSL set of projects on the Zooniverse platform, and we've got several projects now, and we're collaborating with lots of people. And actually, while we still need to get large amounts of these manual annotations to do deep learning, then we found that's a really good way of getting large quantities of annotations to train these networks. And it's worked very well so far, and we're looking forward to lots more exciting things from that project.
And so what do you think will be the most exciting things in microscopy in the next few years? Because like I noticed that as well, equipment wise, even in the last 3 or 4 years, for example, cameras have become very close to perfect, optics have become very close to perfect, and they reach this kind of quantum limits, like you can't have more than 100% efficiency, you can't have less than zero noise. So in the evolution of microscopy, where do you see it going, both technically and like data analysis wise?
Yeah, so I think, you're right. The certain limits appear to be being reached, although people are very inventive and find clever ways of getting around these things. So, for example, super resolution microscopy, which got around the diffraction limit, which was this hard limit that you couldn't beat before. But there's a lot of interesting stuff going on in kind of multimodal imaging. So I mentioned earlier this microscope within a microscope that I sort of got me into joining the EM team. That's a particular type of this multimodal imaging called correlative light and electron microscopy. And the idea is that each imaging method, so it might be X-ray or fluorescence microscopy or super resolution or electron microscopy or histopathology, they all give you slightly different information. So for the example of fluorescence and electron microscopy, in fluorescence microscopy, I might label only the things I'm interested in. So the mitochondria and the nucleus and endoplasmic reticulum, and then those things glow in whichever color I've tagged on them. So maybe using green fluorescent protein and things like that. And then everything else is dark, so you don't see anything else. So you can kind of pick out very specific functions. So you can tell, this green color is associated with something that's performing this function. So I know the function of the thing that I see, but you're limited in resolution. So without going into super resolution and so on, the diffraction limit limits you to a couple of 100 nanometers, something like that, of these fluorescent images. And a lot of the interesting stuff happens below that scale. So you can become resolution limited in that sort of imaging. In electron microscopy, we can now image huge amounts of data. Excuse me. Maybe a single cell might be, if you image it at the highest possible resolution, it might be a terabyte of data, and it might take you, some days to
a
image.
Single cell.
Yeah. So if you're imaging you can image, so for example, something like a focused ion beam scanning electron microscope can image at a few nanometers isotropic resolution per voxel, and that's amazing, right? You see almost everything that you might be interested in seeing, but you also see everything, right? So there's no kind of specificity in electron microscopy. You maybe have some heavy metal staining, so the membranes stand out, but every membrane will stand out. So you'll see these images, and it's a grayscale image, and you've got a whole bunch of membranes. And if you're not an expert, you don't know what's what, and it's very hard to tell things apart. And even if you are an expert, there are certain structures which you can't really identify just by looking at them. So you need extra information. And so combining the fluorescence microscopy and the electron microscopy, you can use these sort of somehow complementary sets of information. So you can tell from the fluorescence microscopy, the thing I'm interested in is over there. So maybe I can save a bit of data on my electron microscope by only imaging over there where I know that object is. Or you can identify things that you couldn't identify in your electron microscopy. And so that's just two imaging modalities, but we also have X-ray microscopy, or it's also known as micro CT, where we can do kind of micron resolution imaging, but over centimeter scales. And then that can be linked to all sorts of other types of imaging, right? And so these things, they all give you different information, and combining them in a smart way, I think, is going to be one of the next revolutions in how this imaging is done. Because as we've seen, if you just blindly acquire all the data that you can possibly acquire, you have too much data and you can't analyze it. So you need to somehow be a bit more nuanced in how you get the data, and then how you use what data you have in the analysis.
And so what we saw, actually, one of our first customers was that they were interested in correlating the two aspects of spectrum and shape, so that if you had some multimodal imaging, what you could then try and do, and in that case it worked well, was to train with that multimodal data your machine learning network, and then to only by an inference from just one of the two modes, say just a shape or just the spectrum, what the other one would be. So, for example, you would get from the fluorescence, you could get a super resolution kind of generative image, or vice versa, from a very high resolution electron microscope image, you could try and infer what the function is, what fluorescence that thing would have. So is there anything going in that direction?
Yeah, so I think the combination of AI and multimodal imaging is super interesting, because AI, deep learning methods sort of associated with the whole kind of big data movement, a lot of that is this idea of bringing together disparate types of data and recognizing patterns across things that maybe a human can't recognize patterns across, because maybe there's too much data, or the different data sets are so different that it's hard to sort of see how everything fits together. But as purely an optimization, multi-dimensional optimization thing, then they're just numbers to the machine. And yeah, I think that's a really interesting area, and I think there's definitely people exploring all sorts of these methods. So that the generative methods, for example, or the sort of mapping methods from one data set to another to try and shortcut having to do certain things. There are certain imaging steps which are maybe very tricky to do, but if you can infer with a good confidence, again, it can be quite hard to convince a reviewer too that that is a robust thing to do, and you're not just producing artifacts, because a network that's trained to predict certain things can predict them inappropriately very easily. So if you Google around some of the types of errors that generative methods can make, they can be quite amusing, but also a bit worrying and hard to extract. So this all needs to be coupled with a really sort of thorough analysis of how we're confident that this is a meaningful thing to predict. And often it is, and you just want to be belt and braces on making sure that there's no possible holes that might come back and bite you in the future.
Yes, I saw that there's quite a lot of work in that direction, and also we try to understand better how those things work, and especially if your kind of input space doesn't, that you use during inference doesn't match statistically the one that you use during training, then if you get the wrong image effectively, then you can have some errors, because the network's not used to seeing that type of image. And there's still very little technology to characterize what space this input actually lives in. And actually, there's, I think, quite a big parallel with quantum physics, because in quantum physics, you have all these like hugely multi-dimensional spaces, where the dimensions are not necessarily orthogonal, and you're always trying to find what is the best representation of that space. And in machine learning, like if you look at that input space as well, it's very interesting to know what the real underlying dimensions are and if they are orthogonal or not.
Yeah, I think that's one of the things, again, having come from the sort of quantum physics training, then you get used to throwing around these multi-dimensional things, and you sort of you sort of come to accept them. Whether you understand them or not is another matter, but you get used to it, and actually it's conceptually a pretty hard thing. And when somebody's just faced with okay, this problem has this many dimensions, then it can be hard to interpret that, or at least to deal with that. And so I think that's one thing that the sort of training we had.
And in experimental physics, it helps that you have an actual physical thing in the lab and is entangled, and then you see what happens, and then suddenly you get an intuition for what those dimensions actually are. But it's true that it's not always obvious. So I also have, I think we're nearly out of time, but I have the bonus question that we have is, if you think in the next few years, now, and that question is much more general, not just about science, but in general. What do you think? What do you think the main theme that will define society will be?
Yeah, that's a good question. So I think, I suppose both in the science that I do and more generally, I think this idea of integrating things across boundaries, I think, is super important. You know, things, because of industries and so on, things get very siloed, and so development happens behind closed doors in certain things, and actually, there's probably people within these silos all solving the same problem over and over again, and just some sort of integration and communication between those separate fields, I think, is really powerful, and we see it in the sort of analogy of this correlative light electron microscopy. It's, you get something that's more than the sum of the parts when you combine them, and I think that is a that feels like something that could happen more generally, right? That there are things going on that for whatever reasons people are kind of keeping hidden, and people have profit margins and so on to look after, but actually, if the purpose is to make life better for more people, then it kind of seems that somehow combining these things, these knowledge bases, would help that.
Yes, I indeed, like it's I think that's just right. Like with, for example, file formats, like there's always a lot of different initiatives, and ultimately it's more effort for everyone if one doesn't work with everyone else to like solve these problems. But yeah, that's really great insight. So yeah, we'll try and be we'll try and be as open as possible as we are, and so yeah, well, thanks a lot, and yeah, well, I will see you soon, hopefully. Like, here things have reopened, I think, in the UK as well.
Yeah, good to see you again, Bruno.
Yeah, so yeah, have fun.
See you soon.
Bye. Bye.